Good afternoon. My name is Ben Greenfield. I'm the Director of Marketing at Helmer Scientific. I would like to welcome you to today's webinar titled Options for Pre-Transfusion Testing, Where Does Tube Testing Fit? Please feel free to submit questions during the webinar using the questions pane. We will have time to answer questions at the conclusion of the presentation. All slides are posted for download in the webinar software. The link is listed above the questions pane on your screen. We will also send a follow-up email that will include a link to this presentation. I would also like to remind everyone that this program includes 1 hour of PACE credits for participants. We appreciate the Blood Center of Wisconsin, a member of the Versity Group, supporting this continuing education program. After the webinar, you will receive a follow-up email with a link to an evaluation form. Completing the evaluation will automatically submit your information for PACE credit. I am very pleased to introduce today's speaker, Sue Johnson. Sue Johnson is Director of Clinical Education and the Specialist in Blood Banking Program at Blood Center of Wisconsin, part of the Versity system. She's also Director of the Transfusion Medicine Program at Marquette University and Associate Director of the Indian Immunohematology Initiative at the clinical instructor in the College of Health Sciences at the University of Wisconsin-Madison. Sue has 25 years of experience in immunohematology reference laboratories and developed interest and expertise in autoimmune hemolytic anemia. Susan shares her expertise through lectures at both national and international educational programs. We are thrilled to have Sue as our speaker today. Without further delay, I'm pleased to turn the presentation over to Sue Johnson. Thanks, Sue.
Thank you, Ben, for that kind introduction. And thank you also to Colleen and the group from Helmer for the invitation to do this webinar today. I always love talking about pre-transfusion testing, and especially the tube testing piece of it, right? Because you'll see as I go through today that I'm still a strong believer that you need to have test tubes available for much of your pre-transfusion testing. So objectives for today, I'll be reviewing the technologies available for pre-transfusion testing, and of course looking at advantages and limitations of those different techniques, and then talk about some different scenarios where it makes sense to utilize test tube methods. I'm a firm believer, I love all the methodologies available, but we have to be cognizant of what they offer to us and where there might be places where we need to do a little bit more testing or problem solving to solve those kinds of problems that we might run into. So let's start with ABO-RH typing. If we look at it from just the methodology perspective, of course, we have our tried and true test tube method, and we know that it's quick. We can do it usually in under 10 minutes. It's been the gold standard, right, for ABO typing for years. We also have available to us microplate testing. Now, if we look at our different methodologies that we have available, some of the automation actually, for example, the automation that has solid phase testing actually utilizes a fluid-based ABO type method, which is the microplate method. So the antisera, anti-A, anti-B, anti-D is added to the well instead of a test tube, and then incubated, agitated, and then read. And then finally, we have column agglutination technology that's available, either gel or glass beads outside the US. And again, really great methodology for detecting and to determining ABO type. So it's great, we have options. But one of the challenges is when we have an ABO discrepancy, and I think that's where I see tube testing absolutely fits in. So if we just look quickly at reviewing an ABO discrepancy, right, we all know the standard is that if the forward doesn't match the reverse type, where we should see an A, if we see an A in the forward type, the reverses should also reverse as an A, meaning that the patient has anti-B. If those don't correlate, of course, there's a discrepancy. We need to go into a problem-solving mode. Another discrepancy, ABO discrepancy, is where our present type what we just did, what we saw in the testing, is not matching what we have in our history. So we have a record now of a patient that is an O, but we just typed them as an A, for example. So that is definitely, I would consider a discrepancy, although not by the classic forward doesn't equal reverse definition. And finally, also, I like to classify as an ABO discrepancy those results that don't meet the expected results. So for example, when we do ABO typing, we like to see 3 to 4 plus positive reactions. And if we see only a weak positive, or we see mixed field reactions, that is important to note because that gives us information about the patient. And maybe they had a stem cell transplant. Maybe it's a subgroup. There's lots of different scenarios where unusual reactivity should flag that this is a discrepancy of sorts. Now, that's, I think, where the challenge is for some of the different methodologies. So for example, if we look at a patient that has mixed field 2 cell populations, for example, a group A patient who's received a group O hematopoietic stem cell transplant, or what we used to call a bone marrow transplant, the instrument might actually call it as a question mark or an NTD or no type determined. So what it's calling us to do, right, as a laboratory scientist, is to look at that and try to determine what is it, right? We could visually look at it, but I think that's difficult to do on that little microplate well, right? So a place where the tube testing can really fit in is to in this kind of scenario is if we repeat the testing where we have this no type determined or question mark in a tube, we could see, there's nice mixed field here. There's a classic, in this case, 4+1 agglutinate with lots of free cells in the background. And then it totally makes sense with our patient history. Patient was an A, we're seeing their A cells, but they're developing into an O, so they're starting to produce their O cells, and thus we see that cloudy background. So those are the O cells that are unagglutinated. So classic example of where our test tube method could absolutely show us that. Now, for you column users, you know also that mixed field will also show very nicely in a column, in a gel test or a column agglutination test with beads. You'll see the agglutinated line and the unagglutinated line, or the unagglutinated cells that fall to the bottom. So I just don't have an image of it to show you, but gel would also provide that information for you. Another scenario where we might see maybe unusual question mark or no type determined type reactivity is when we have missing or weak antigen. So for example here, if we see this, it might be suspicious. They back type as an AB, they forward type with a questionable reactivity with anti-A. I'd be thinking it's weak A antigen based on the back type, right? So I think the back type is correct. So something is unusual is going on with the typing with the anti-A. So again, if we can go to a test tube method, sometimes all it takes is a little bit different methodology. And when we do a test tube method, of course, we're centrifuging, and that sometimes is just enough to give you a nice, stronger positive where you feel confident in the typing that, yes, this weak A antigen is actually weak, I agree, but now it's within that realm of, yep, this patient looks like an AB. There's no question mark there anymore. And then another place where it can become helpful, and when we look at tubes versus other methods, is one of the things we know about column agglutination is that it is not the best at detecting anti-A and anti-B. I mean, we just know that about the method. And so it's not surprising to see 2+ reactions with, say, for example, B cells. Now, in this example, I'm only showing a weak positive, but 2+ is not that unusual. But when you get a weak positive, that makes you question it, right? Or even the instrument may question that. So again, we know this to be true. And gel is wonderful, a column is wonderful for lots of things, but for ABO, it's sometimes that's one of the limitations of the method. That if you go ahead and repeat it in a test tube method, just a regular test tube method with A cells and B cells, now you see that the B cells are a 3+. And we feel much better about that reaction. And we can now say, yep, my patient is an A, and I really don't have an ABO discrepancy. So it's just another place, again, where I think test tubes can really be valuable in solving those kinds of issues. And then what about this example? Here is another ABO discrepancy, and now it looks like we have what we might say extra antibody. So if I take a look at this, what you can see here is we're forward typing as a B, and reverse typing as an O. But the thing is that these B cells are 2+. So forward as a B, reverse as a O. That means I have a discrepancy, obviously. So what do I think is correct? I usually look at where I have a clear negative and a strong positive. So I believe this is correct. My patient's really a B. So my extra reactivity, the extra antibodies with these B cells. Now you look at your antibody detection test or your antibody screen, and it was performed in either column or solid phase, and it's negative. And then you're like, okay, looks like I have extra reactivity here, and there's no reactivity here. But what we have to remember is that our antibody detection methods, especially column or solid phase, are designed to detect IgG antibody. It may not detect an immediate spin antibody that is reacting unexpectedly with these B cells. So again, if we go to a test tube method, that allows us to look at different phases of reactivity. And you could see now here, and when we run our panel, you could see that there's nice reactivity at immediate spin, there's reactivity at 37 degrees, and I know all the blood bankers are like trying to eyeball that and figure that out, right? But what it looks like is, a nice anti-M, because all the M positive cells are positive and the M negative cell is negative. So this just is a nice example to show that the different methodologies are wonderful for IgG antibodies. But if we have an ABO discrepancy due to an IgM, we may not see it until we go to using a test tube method, where we can look at immediate spin to identify that IgM antibody. And then, of course, to finish off this case, if we look at our forward type as a B, reverse type as a O, we still have this discrepancy, so we have to do something to resolve that. We could pre-warm, warm the A cells and the B cells at 30 degrees and then centrifugally and read, or we could select cells that are M negative and verify that the reactivity is there only with the A cells, showing that is exactly what we want, and that the B cells are negative. So we can see now that my patient forward types as a B, and back types as a B. So now we have our discrepancy resolved. And again, all this kind of testing most likely is going to be done in test tube methodologies, although you could do this as well as in a column. Okay, so that's ABO. One more thing though, just to touch on with ABO, is what if there's an urgent need for transfusion? Our instrumentation, the different methodologies are wonderful, but they take some time. There's a long centrifugation phase, there's a processing time. So I think here's another situation. If we don't have a historical ABO-RH, the test tube method for ABO-RH typing is much faster, right? Again, like we can do that in 10 minutes, right? Versus waiting for that centrifugation or processing time. So another place where I think tubes definitely have an advantage. All right, on to RH, or typing with anti-D. So again, most of the time when we're doing our testing with different methodologies, it's not a problem. But we all know, we're all challenged by those individuals that have a weak expression of the RHD antigen. And today, I mean, for RHD gene alone, there's greater than 200 alleles that have been identified that cause weak expression of the RHD antigen. There's those individuals that are weak D types, over 135. Those are generally the people that have a quantitative difference. There's much less antigen expressed on the red cells. There's many different types of partial Ds that have been described now, where an individual is missing an epitope of the D protein, so therefore they can make an antibody to that. And then there are the individuals that are very, very weak, that probably no methodology will detect, but definitely part of all these different alleles that can cause weakened expression of the RHD antigen that we're trying to detect with our normal typing. So, you know, we've all seen this if you work in blood bank transfusion service at all. The frequency is estimated to be about 2.9% among a mixed population in the US. So meaning if you have a diverse patient population, you're going to detect about 3% or so of individuals will have this serologic weak D type. If your patient population is mostly of a European background, then it's probably more around 0.5% to 1%. But it is enough, right, to cause consternation and challenges sometimes in our typing. And if we look at how do we define or how do we detect these unusual phenotypes or RHD phenotypes, is usually by definition that was defined in by a work group that came together with the CAP and AABB led by Dr. Sandler, where we defined a serologic weak D phenotype. And that would be an anti-D reagent that gives no or weak reactivity, less than or equal to 2+ in initial testing, but agglutinating moderately or strongly with antiglobulin reagent. Okay, so anti-human globulin is you know, most often, right, applied in a test tubes or column methodology. And the other place a serologic weak D phenotype would be detected is if you have discordant typing results. So you have a history of a patient being RHD positive, for example, and now you're typing them as D negative by whatever methodology you're using. And so those are challenges for us, right? Those are things that we have to deal with. And again, when we look at our different methodologies available, microplate methodology may call it question mark or no type determined, gel or column, again, questionable, or even a less than or equal to 2+, you could consider that a questionable RHD typing. And again, if it was discordant with your historical typing, it would be another place where you'd be like, okay, now what? What do I do? Okay. On this slide just shows one example of an RH discrepancy algorithm that I think is used quite often in one form or another. But basically what it's saying here is that if your anti-D is greater than 2+, then you're good. And it matches your history, you're reporting your patient as RH positive. If it's not greater than 2+, then and your standard test anti-D testing, your standard methodology is negative, you repeated it, and you still have inconclusive results, then you would go on to test with different anti-D reagents, definitely at immediate spin, and probably doing an antiglobulin test. It's tedious to do, right? And most of us probably don't have more than 1 or 2 anti-D reagents available, but that will help us in resolving these kinds of serological discrepancies. And in fact, today, at last count, I have a student, we're doing a project, and there are actually 17 different anti-D reagents that are available on the market. Now, some are for gel, some are used on different automation, automated platforms, but there's a lot of different anti-D reagents. And for the most part, most of them can be used in tube testing as well. And in fact, there's one paper that was published actually in May of this year where they actually used that to their advantage to determine when would we consider doing RHD genotyping, for example. And in this particular paper, I think just to point out that they used a gel anti-D and a test tube anti-D. And so they looked at this as a clue for when to do molecular typing. And you can see here they talk about if the gel was 2+ or greater than the test tube result, or was less than or equal to 2+ with both methods, or less than or equal to 2+ with gel, or less than 1+ with test tubes only, or there was an anti-D, they would actually go ahead to do RHD genotyping. And interestingly, in the 49 patients that they tested, or in 49 patients, they saw 39 partial Ds and 10 weak D types. So using both test tubes and gel, they were able to determine those individuals that we should call Rh positive, the weak D types most likely, or Rh negative, and treat them as Rh negative, which would be the partial D types. So again, just showing the value of where test tubes could come into play. And of course, all of our the weak D tests that are done along with it. Okay, moving on to our antibody detection test, or antibody screen, as we all call it. Here's, where there's actually lots of different possibilities. We all know that we want our antibody screen to detect antibodies to red cell antigens in an individual, in most cases our patient, but also in our blood donors. We know it has to detect IgG antibody, that's the standards. And it might detect IgM antibody. And I gave you that example showing where we had negative antibody screen, but we detected the IgM in the ABO type. And of course, our goal is to detect as many clinically significant antibodies as possible while minimizing the detection of insignificant antibodies. Always our goal, because we don't want to get those extra positives that we don't know what to do with. So if we look at our different methodologies, of course, our first, the gold standard, I still think, is our test tube methods. Of course, that's my opinion. But we've done lots of things over the years with our test tube methods, right, to ensure that we're detecting antibodies. Our standard is a 2-to-1, 2 drops of serum to 1 drop of cells, but we know we can enhance our test tube method by adding more drops of serum or plasma. We can incubate our incubation time, we can increase our incubation time from say 30 minutes to 60 minutes. We have additives, and there's benefits to using all of these different additives. LIST increases the speed of antibody uptake, so it doesn't take as long to do the test. PEG increases antibody uptake and also agglutination. And enzymes, we all know, testing enzyme-treated cells will give us lots of different hints, right, as to is the antigen destroyed to which the antibody is specific for, and we can sort things out using those. And of course, when we're looking at detecting antibodies, we always want to have red cells that have a nice double dose or homozygous expression of the antigen as much as possible so that we can detect all antibodies. And, we all know, and of course, all of these methodologies, again, we like tubes because we can read at immediate spin, we can read at 37 degrees, and we also know we like to be able to do our antiglobulin testing. And of course, there's actually some wonderful cell washers available now. One pictured here that I've had the opportunity to use, and they're really cool compared to the old, like my original cell washer was a green looked like R2D2, and actually I have the rotor on my desk as a pencil holder right now. So some of you can visualize this, I know. So the new cell washers are pretty slick. And of course, it will give you nice results. So when we're doing tube testing, it's important to remember then, right, if we read out all the phases, this slide just lists all the antibodies, IgM type antibodies that we would see most likely at immediate spin or room temperature. And remember that there are antibodies that will react best or maybe only after the 37-degree incubation and spin time. And that's usually IgM type antibodies. We'll go through that more in a bit. And then you come to your antiglobulin test, and of course you see everything, all of the IgGs, and including anti-CD38 or DARA, which is pretty a popular topic right now. But again, we can see all these different antibodies reacting at these different phases. Now, if we move on to column agglutination, as I mentioned, it can be gel or beads. Just have two listed or shown here on this slide. One has got 3 columns, and the other has 6 columns. Can't count. The other has 8. But these are nice antibody detection methods where they utilize lists. So the red cells are suspended in a list solution. And then incorporated into the screen, into the cards, is an anti-IgG. So these methods absolutely will detect IgG antibody. So of course, we know it's automatable. It's a standardized method. We don't have to wash like we do with our tube tests, but we do need special incubators and centrifuges and pipettes. And as I've alluded to before, there's a long centrifugation time, about 10 minutes for most of the methods that are available, or I should say exactly 10 minutes, depending on the manufacturer's directions for use. There are some other unique characteristics just in reactivity. So cold reactive antibodies may show that mixed cell or mixed field agglutination. So here is a classic mixed field type reactivity where you see the agglutinated cells at the top, unagglutinated cells at the bottom. If you're doing an ABO type, you would say, as the example I showed you, you would say that's 2 cell populations. It's an A and an O, for example. But if you're doing an antibody screen, you shouldn't be seeing mixed field per se by the true definition. There aren't really 2 cell populations in your screening cells, at least you hope not. So if you see that kind of reactivity, that's actually characteristic also of a cold agglutinin type antibody. Could be an anti-M, a cold autoantibody. And what you see is that the antibody, the IgM antibody, probably binds right away when you mix the plasma with the red cells. And then during that 37-degree incubation, some of the antibody falls off and falls to the bottom of the microtube. So you actually get this kind of characteristic agglutination, and it can give you hints as to what the antibody is. And that versus rouleau, which kind of is more stringy. You kind of see the cells kind of hung up in the column versus here where it's a little bit more clear cut. Cells at the top, cells at the bottom. So it's a nice clue when you see that kind of reactivity. Other unique characteristics, we know column agglutination is wonderful at detecting anti-D. There are some other antibodies that have been reported to be weaker, anti-big E and anti-big K, and in more previous days. I haven't heard that as much now. And it's not as sensitive at detecting anti-JKA and anti-JKV, especially as compared to solid phase methods. We know it enhances warm autoantibodies, and it may not detect the agglutinating antibodies, the IgM types. And of course, and I say that of course because we always have that, there are some unidentified positive reactions, and you don't see hemolysis as quite as much. And speaking of those unidentified type reactions, one of the classic reports came out in 2013 by Liu and Grossman, where they looked at the antibodies of undetermined specificity, 30 months of antibody detection tests, or over 138,000 patients. And they found that 18%, or over 1,400 tests, had undetermined specificity. So positive reaction where they weren't able to identify an antibody. And most of these were weak to 1+, and actually were the most reported reason for a positive antibody screen. And when they went to the PEG test tube method, they showed no antibody present. And interestingly, 7 developed into new antibodies. So definitely there's antibodies of undetermined specificity in using the gel methodology. Now, as far as looking at the serologic behavior of antibodies, I showed this slide previously, looking at test tube methods. And for column, it's similar. If you're using the buffered card, you'd be able to see the IgM antibodies. If you use your buffer card and incubate it at 37 degrees, you'd also see these IgM type antibodies, and as well as, of course, all the IgG antibodies. Now, the thing is, most of us don't use the buffered cards routinely. So you have to remember that, again, if you aren't looking, you won't see some of these IgM antibodies. And most of the time, that's fine. We don't care, except for, of course, like I showed in that ABO discrepancy example. Moving on to solid phase red cell adherence. This methodology is totally different. It's working with a micro well where red cell stroma of the screening cells is actually effaced to the well, and you really can't see it with the bare eye. And so what you do is you add your patient. This is a negative patient antibody in this column, and you can see that if the antibody is specific for an antigen on the stroma, it'll bind. And the way that we visualize it is by adding IgG-coded indicator red cells that will bind to the patient antibody. So a positive, the cells are all effaced, and negative, all the cells fall to the bottom. The other solid phase methodology is one where protein A is coated on the well, and it's here we actually add directly a drop of patient plasma, a drop of the screening cells, and some lists. Patient antibody will bind to the red cells. You wash the well, add anti-IgG, and then again, the IgG can preferentially, because of the pH of the buffer, can bind to the protein A, and then the antibody-coated cells, screening cells, will then bind, and again, a positive will be all effaced, and a negative, the cells will all fall to the bottom. Again, it's automatable, standardized method, but of course you need special incubators, centrifuges, etc. Absolutely, solid phase methods are enhanced anti-D. Been nice reports, and I think almost everybody that has used solid phase red cell adherence has seen an increased detection of anti-JKA and anti-JKB. Sensitivity is high for all antibodies. Warm autos, of course, also are enhanced, unfortunately. There's a little bit less variability in a positive reaction. So for those of us that are used to solving antibodies and looking at for variability and positive, like one's a 1+ and one's a 3+, you don't see that quite as much using the solid phase methods. You may not detect agglutinating antibodies, those IgM types, probably less so even than with column. You just wouldn't see that. But there's an increased rate of unidentified positives. So of course, with the increased sensitivity also comes a decrease a bit in specificity. And the other thing that's unique is that few people test the autocontrol because you have to prepare your own monolayer of the patient red cells on the bottom of the well. And now, what about antibodies of unidentified specificity in solid phase red cell adherence? This is a paper that that we did at my facility, and we looked at 6 years of antibody detection tests. So 4 years of using a PEG tube method versus 2 years of using the solid phase red cell adherence. And Unidentified specificity was reported using PEG tube as well, but only at 1.8 per 10,000 tests versus using solid phase where it was we calculated at 20 per 10,000 tests. So definitely more unidentified specificities. Now, just like with column, we have to remember that we're only looking for IgG antibody. And so again, these are the antibodies that we would most likely see if we're using a solid phase method. Now, when would you use tube tests, regardless, right? When you're using gel or solid phase? Well, one is inconclusive results, right? We switch methods. We look for a way to determine, is this real reactivity? Is it an antibody we need to worry about? We'll also switch methods when we look where we see cells where the patient reacts with almost every single panel cell we test in the method that we're using, solid phase or gel or beads. Or if there's an IgM antibody we suspect, or even a newly developing IgM antibody. We'll look at each of these a little bit more. So one of the things that both reports that I talked to you about, Lou or Miller, they looked at what about transitioning of unidentified reactivity to real antibody. And you can, interestingly, when you compare the two, gel showed 31% of these unidentifieds transition to an allo or an auto. In the PEG-2 method, 29% transition to an allo or an auto. And the solid phase method, only 16%, which again shows us that it's a more sensitive method, but also more sensitive to interfering substances. So again, leading us to looking at having to do some tube testing or another methodology, or could be gel. Gel or solid phase, positive with most red cells. Again, in most cases, if everything's positive, you're going to be going to rare cells, testing antigen negative selected cells, and tube tests are generally where we're using what we're using for those kinds of methods, although you could again use column. Warm autoantibody, you have a patient that has a strong positive DAT, the panel's reactive with everything. We all, like I already mentioned, both solid phase and column is good at detecting warm autoantibodies. So many people in the IRLs like to go to a saline or a Lys antiglobulin test because it's less sensitive. It's still a good method. It'll detect alloantibody, but it won't detect the warm autoantibody. Cold autoantibody, kind of similar, except here now it's reactive with a strong positive DAT, may or may not be positive in your column or gel, but we know that if we do immediate spin and test tubes, we're going to get a good handle on what's causing the positive in the other methodology. In anti-CD38 or DARA, or the other drugs now that are on the market, there are some labs that are taking the approach that if it's positive in my automation, I'm going to use my test tubes next to see if the antibody is still reactive. Again, the test tube methods, kind of have them in the order here, saline, Lys, PEG. They're least sensitive or less sensitive to the anti-CD38. So you may actually have a negative screen, which is great because then you can still rule out alloantibodies. And then what about those inconclusive antibody IDs? Remember, your IgM antibody, if it's there, is going to react best at immediate spin. Your 37-degree reactive alloantibody could be a newly forming antibody that's just transitioning from IgM to IgG. And again, this allows us to test different phases of reactivity. So as an example, of course, here's another antibody that reacts best at immediate spin. Incubation, it gets a little bit weaker, but there is one positive in the antiglobulin test. And if you look closely at this one, this is consistent with an anti-P1, which we know classically can vary greatly in reactivity between different donors. And you will get that unusual result, but you wouldn't see it by doing just an antiglobulin test. So here's a nice anti-P1 that's reactive at immediate spin and 37 degrees. And then, as I mentioned, there's those antibodies that are just being developed, the primary, the patient's been exposed. And we all have to remember that if a patient's been transfused and it's within, say, 10 to 14 days, it could be the primary immune response. And the first antibody that shows up is the IgM class antibody. So if we catch a patient and we're doing our testing at the right time, that is where we might see the best reactivity is right here when they're developing the antibody. And what that can look like, if we go to we might see some weak positive that doesn't make sense in whatever method that we're using. But if we go to do a test tube method and read for agglutination at 37 degrees, we might see perfect reactivity. And in this case, classically, anti-Big E is famous for doing that. The other antibody that potentially is kind of famous for doing that is anti-Big K. And again, it might be one that we wouldn't see or wouldn't be able to figure out until we tested at that phase of reactivity. Okay, that's all about antibody screens and antibody detection and where test tubes can come in handy. Another place is when we're doing our cross-matching, right? Using cross-match, you know, most of us use an immediate spin cross-match, or an electronic cross-match is probably the best methodology for a cross-match. But you might come across a case where you're doing test tube immediate spin cross-match, where you've done either column or solid phase and you have a negative screen, but your cross-match is incompatible. Again, we're testing it at a different phase of reactivity, and actually we're looking right in a cross-match for ABO incompatibility, so we should expect to see reactivity with a method that'll detect an IgM antibody at immediate spin. Now, this incompatible cross-match, right, can be due to an ABO incompatibility. Of course, we're all going to check that right away. But again, it could be due to an IgM antibody, could be an antibody to a low prevalence antigen, could be low, probably not a 3+, but it's possible. Again, we're not going to see that though unless we look at it in a test tube method, or the donor has a positive DAT. Least likely with an immediate spin positive, but all could cause an incompatible cross-match. So just something to remember. I'm not going to show you another panel, but just remember that we need to, again, be able to do some immediate spin testing or a method to detect that IgM antibody to determine why we have that incompatible cross-match. Moving on to our direct antiglobulin test, another place where we consider using tubes. So in places that, of course, normally you would do an auto-control when you do an antibody identification panel, especially when you're doing test tube methods. And if you're switching methods, we always like to include an auto-control. We'll do a DAT when the physician orders it. It could be generally a column agglutination test, gel test, or beads, or a test tube method. And if you don't have column, you're probably doing a test tube method. And the other place that we'll be doing our direct antiglobulin test, mostly in test tubes, is when we're doing a solid phase antibody ID panel. Again, I mentioned that we don't normally, most people don't run an auto control just because of having to prepare that monolayer of cells. So they'll go to doing a test tube DAT, or you could do a column as well, but an alternative way to do the DAT, and mostly in test tubes. And of course, if your DAT is positive, we're going to have to test our eluate. And again, if you're doing an old Louis freeze-thaw eluate where you are looking for ABO antibody, you lyse the red cells by putting them in the refrigerator or freezer. You freeze the red cells and then you thaw them slowly and you lyse them, and the eluate is very hemoglobin stained. So it'd be very difficult to read unless you're using test tubes and reading after you wash the cell. You do an indirect antiglobulin test. So you're going to wash the reagent cells with the eluate, wash away most of that hemolyzed eluate, and add anti-IgG so you can read in a test tube method. Rapid acid eluate, test tubes the preferred method according to manufacturer's directions, but you can validate for use in solid phase and solid phase methodologies. But I think most will use test tube methods if you're using or performing a rapid acid eluate. So in summary, we went through a lot. We went through a lot of different possibilities or opportunities for using test tube methods. We went through the technologies available for pre-transfusion testing, including the column agglutination testing, the solid phase methodologies, and this solid phase methodologies being a microplate methodology. We went through all the antibody detection methods, talked about crossmatches and DATs. We looked at the advantages and limitations of the different methods, where we could see, yeah, these are definitely, this is an advantage or something I can see that's unique to this method, as well as the limitations to those methods. And it gave you a lot of different scenarios of where it makes sense to utilize test tube methods to help either in the initial testing or when we go to problem solving. And with that, I'd like to say thank you for listening, and I'm going to turn it back over to Ben for some questions.
Thank you, Sue. Thanks for sharing your presentation today with our audience. We are now ready to move to questions and answers. As a reminder for all our participants, you can submit your questions using the questions pane on your screen. If we don't have time for all submitted questions, we'll make sure to follow up after the webinar. Again, please use the questions pane on your screen to submit your question. So Sue, the first question that we have is, my lab uses mostly generalists. Is there still a place for tube tests in the transfusion services?
Great question. So I think I gave a lot of different scenarios where there is the value in the test tube methodologies. I guess it would depend on the your patient population and what what your general patient population is, and how often you would come into the scenarios that I talked about. I think there's always value though. I know many labs will, especially a small laboratory where they have a lot of generalists, might send to another hospital, for example, in their system to do additional work. But the one thing you always have to remember is that there's always then a turnaround time issue. And if it's something that can be quickly resolved with a test tube method, maybe there is that value to have that. And I know a lot of people say, but I have to keep my laboratory scientists trained in using the test tube methods, and yes, you do. I believe you do. And I am happy to report that at least all the schools that I know of still teach the students, our seniors learning test tube methodology so that they're ready to be able to troubleshoot and problem solve.
Thanks, Sue. The next question that came in is, have you dealt with DTT treatment of anti-CD38 drug-coated cells?
Yes. Well, drug-coated cells are testing in the presence of, DTT treated. Okay. So, yes, anti-CD38 is causing trouble for everybody. And in our laboratory, we actually use AET treated red cells, and it's validated to test in test tubes. So it works well. I mean, it works well. I have heard some that are validating it to use in column agglutination.
Great. The next question, the question is, what PEG manufacturer do you use at your lab, or what do you recommend?
Okay. So I don't usually recommend a manufacturer. However, this is an easy question for me because we actually make our own. We prepare PEG in-house, and we use, it's a 20% PEG solution that's made, actually the procedures from the Judd's Methods book. So it's really easy and straightforward and inexpensive. but you have to do the validation and testing to, just to verify that it works, all the quality control to verify that it works appropriately.
Thank you. The next question is, what is your current recommended protocol for unspecified antibodies on SP DAT negative?
Good question. So based on the study that I showed you, by Miller et al., my thought is, and this is now my opinion, but it's also based on the study, is that I would go to a PEG tube method and see if you see any kind of reactivity at all. If the PEG tube method is negative, I am perfectly fine, and I've heard many people now saying that that's the methodology that they feel comfortable with, that if there's no antibody there in PEG, we're not worrying about it.
Great. Let's see, the next question is, between the LIS and the PEG method, is there one you feel that is more sensitive or preferred?
Yeah, you guys are asking me great questions. So I totally, 100% prefer PEG. The reason is that if you look even at the data and look at studies and just my personal experience over 20 years of using it, PEG enhances agglutination. It'll take an antibody that's, say, a weak positive in LIS or saline and make it a 2+. I mean, it'll enhance agglutination at least one reaction grade of true antibodies. Just a little background on that. LIS was designed to, even though it's called an enhancement solution, it's not really an enhancement of agglutination. It enhances antibody uptake because by removing the ions from around the red cell, it allows antibody to get to the red cell faster. But if you go back when LIS was first introduced, everybody was so excited about this and the manufacturers said you can do this in 10-minute incubation. We were all using 30-minute incubation. So everybody's like, yes, I want 10-minute incubation. So we all went to 10 or 15-minute incubation. So that's what we use, but that doesn't really enhance the agglutination reaction. Whereas PEG enhances the agglutination, and the in-house prepared PEG is a 15-minute incubation. It's great. Or if you use the commercial PEG, it's actually a combination of LIS. It's PEG diluted in LIS, actually. So you actually get the benefits of both. So by far PEG. Sorry, that was a long answer, but I wanted to explain why I believe that.
Thank you. We have one more question that came in. It's a longer question.
Okay.
When there is a complex patient population, for example, mixes of hematopoietic cell transplants and sickle cell patients with multiple antibodies, is it always best to keep the tube method as a backup? And the second part of the question is, what type of methodology would be best in this situation based on your opinion?
Okay. So, yeah, complex patient population, as you described, the HPC, the transplants, that's where I think you're going to see more problems or, questionable NTD types in your ABO typing, for example, with that automated platform. So there, I think test tubes would be super valuable to be able to, just, have confidence in the ABO type. The other place with complex patients is with the sickle cell patients and lots of antibodies. I think you want to have test tubes available to be able to sort out those antibody problems, like the examples I gave, where you might have a newly developing antibody, an IgM type antibody, an autoantibody. Many of those multiply transfused patients with sickle cell have developed autoantibody on top of their alloantibody, and one of the automated platforms probably will just call it all positive, and so you'll need to back off a bit in sensitivity to a test tube method. And from a test tube method perspective, I would have PEG, just because I know I believe that it would really give you a nice positive reaction if it's a weak antibody that you're trying to detect. And then actually I would have the ability to just do a saline indirect antiglobulin test, just a no additive, just in case it's an autoantibody that you don't want to detect. Or you could use LIST, but if you didn't want to have the extra cost, you could just go to a saline method. 30-minute incubation, no additive, and then wash and add your anti-human globulin.
Great. Well, that was our final question, Sue. So thank you again for the fantastic presentation and for all the answers to the questions that came in. I would like to thank everyone on the line for participating in our webinar this afternoon. And Sue, if you could advance to the next slide, please.
Sure. There we go.
Thank you. Helmer Scientific welcomes everyone's input on this program, and we actually like to get that feedback to help with future educational events. If you have feedback or comments you'd like to share about our webinar program, please feel free to reach out to me, Ben Greenfield, at bgreenfield@helmerinc.com. As I mentioned earlier, this program does include 1 hour of PACE credit for all participants. After the webinar, you'll receive a follow-up email with a link to an evaluation form. Completing that evaluation form will automatically submit your information for PACE credit. We hope to follow up on our event today with additional webinars with topics based on your feedback. This concludes today's program. Thanks again, and thanks to you, Sue.
You're welcome. Thank you.
All right. Thanks, everyone.